All right, welcome back to the AP Bio series. Today I'm going to be doing a full Unit 4 review of AP Bio and a little bit of Unit 5. So let's first start with some notes for review. So these are really important concepts that you should have learned throughout the unit, but let's just review them once. So firstly, we'll discuss the types of cell communication. Now, when you talk about cell communication, there's a lot of ways that cells communicate throughout our body to send signals. Firstly, there's physical communication, which only happens through physical and direct cell-to-cell contact. So if the cells aren't within a close proximity of each other, then physical communication or direct cell-to-cell contact isn't possible. Therefore, it cannot be physical communication that they're using to send a signal. It must be some other sort of communication. Now, secondly, we have long-distance communication, and this is most likely the case for different cells in our body as well. So this only happens with the use of chemical messengers for cells to communicate with each other. And an example can be seen as with neurotransmitters, where these chemicals go through local cell communication, right, because they directly go through synapses, which obviously is a faster route than physical communication or any other way of communication. Now let's go into some vocab. So a phosphorylation cascade. Now what I want to talk about with this word is just remember a chain reaction, okay? A chain reaction where one enzyme adds a phosphate group to the next protein in the sequence, and so this results in a changed or amplified cellular signal to be released. Now let's learn about signal transduction and the steps that go with it. So steps of signal transduction can be stated as reception, transduction, and response. So those are the three main steps to remember. Firstly, with reception, the target cell receives a signal. So here, when I mean target cell, it's the cell that's receiving the signal, and that usually takes place or participates in this signal transduction process. So secondly, with transduction, what happens is the signal is converted to a specific, easy to trigger cellular response form, which is usually done through transduction pathways. So you can see with an example like acetylcholine or other transmitters or other chemicals as they go through different pathways to be recepted by target cells and to release a response. So the third step in this process is response, obviously. So a cellular response is produced, and that can either make or affect gene expression. So what I mean by gene expression is genes turning on and off. It can lead to, or it can signal to apoptosis, so programmed cell death. It can signal for a cell's death, basically. And it can also signal for the secretion of certain molecules in our body that may be needed or may be necessary at certain states or times. Now here is a simple diagram of the signal transduction process. So first of all, we have this signal molecule right here, which, this first step is reception. So the signal molecule goes and attaches to the receptor protein. Right, there's a little space over there. And what that does is that changes the shape of the receptor protein. And when it goes through the transduction pathway, right, the next step is transduction. When it goes through this transduction pathways and the process, a cellular response is produced as a result. Okay, now let's go into some more vocab. So, G protein coupled receptor. Now this is a type of cell-to-surface protein that when a signal molecule binds, it changes shape and contributes to the production of a response. And it can be cellular, hormonal. There's a lot of different types of responses, so it depends on what the signal molecule signals to the receptor protein or how the receptor protein changes shape within this entire process. But those are some factors here. Now, cyclin. Now, what is cyclin? Cyclin is a specialized protein that when activated with CDK, or cyclin-dependent kinase, this produces a response that informs a cell to progress to the next step in the cell cycle. So, for example, let's learn about cyclin-dependent kinase, so CDK, a type of enzyme that contributes to a cell's progression through the cell cycle only when activated. So it can't really do anything without cyclin. So it's just inactive unless cyclin binds to it or is attached to it in any way, shape, or form. So here's an easy analogy to remember that I use to remember the relationship between cyclin and CDK and their role in the cellular signal transduction process. So here we have a key and a car. Now let's imagine this key right here as cyclin, right? Because here in the car as CDK or cyclin-dependent kinase. Because, well, we can't open the car's doors without a key or even drive a car without the car's keys. And that's the same thing here where we cannot utilize cyclin-dependent kinase, or cyclin-dependent kinase cannot function within the cellular process without cyclin. So the key is cyclin and the car is CDK here. So that's an easy analogy to remember and visualize. Okay, now let's learn about the types of feedback. So there's two types, positive and negative. So positive feedback amplifies a response, so it changes it in some way. It pushes the system at a greater distance than the original starting point to complete a process. So let's look at an example. So we have this example of fruit ripening here. Now this diagram shows an example of positive feedback, right? So what does it do? Well, we have a fruit here, right, an apple. So this apple, when it starts ripening, it produces ethylene, right? So ethylene, this is the chemical composition of ethylene, I guess. It makes other fruits or other surrounding fruits like apples ripen as well to complete this process of fruit ripening. So let's learn about negative feedback. Negative feedback strives to reduce or inhibit a stimulus for the state of homeostasis to be present. So, well, what does that even mean? So let's look at an example. So this example is on childbirth, the process of how a child is born. So first step, the cervix is pressured, and then contractions start, obviously, in the stomach. And then oxytocin is released as a result of or due to increasing pressure within contractions and cervix. Then, with more resulting contractions, pregnancy and recovery happens. Now, with this, we can see that, negative feedback strives to reduce or inhibit. Now, the negative feedback here is striving to reduce or inhibit the pressures that occur within the cervix or the contractions or any of these processes within this big process of childbirth. And so that's why oxytocin is released as a result due to increasing pressure, right? It's trying to inhibit that pressure. And thus, with more contractions, more oxytocin is released. And finally, as a result, pregnancy or a baby is born. So next, let's learn about the steps of mitosis. Now with mitosis, the first step is, interphase. Though there's no diagram here for that. Basically, interphase is 90% of the entire cell cycle, and it consists of 4 kind of steps in this process. So firstly, there is G0, where the cell is kind of resting. So G0, where the cell is at rest. So at rest. Okay, now the cell, you can imagine this as kind of retirement, or that the cell is in a waiting room, because here in G0, the cell is waiting to receive a response to move on to cell division or move on to the cell cycle. Otherwise, the cell is at rest and stays here, no matter what, at the G0 stage. Then there's the G1 stage, which is obviously G stands for growth, so growth 1. In this stage, the cell produces enough proteins and more stuff, we can say, in order for cell division to successfully occur, and for enough proteins and stuff here, quote unquote, is enough basically for the two cells that are produced as a result of mitosis. Now, then after that, there is G2, where, again, proteins are produced, and basically a continuation of G1. Now, then there's the S phase, where synthesis of DNA occurs. So DNA is essentially replicated here. And then, let's see, and then we have, I think that's it, yeah. So G0, G1, G2, and then we have S. So let's learn about the steps of mitosis. First, we have prophase, where we have a cell. We have a kind of centrosomes at opposite poles of the cell right here and right here at the left and right sides, as you can see. The nuclear membrane is starting to disappear, and we see that there are spindle fibers that are also starting to develop in this stage from the use of centrioles, which are only found in animal cells, or centrosomes, which are only found in human or somatic cells. Now, let's move on to metaphase. So in metaphase, we have the same centrioles or centrosomes here with even more developed spindle fibers. So they're developed so much that they can even attach to the centromeres of the chromosomes. So it's important to understand the structure of chromosomes. So we have a chromosome here, okay? Within the structure of this chromosome, we can see that in the middle, this is called a centromere. And this is called a sister chromatid. And this is another sister chromatid. So if you join one sister chromatid and another sister chromatid, this centromere is what connects them together, and that's what makes a chromosome kind of form. So originally what happens is within prophase, whatever was within the nuclear membrane was chromatin. And it's kind of this messy form of DNA, so that has condensed into chromosomes. And the nuclear membrane starts disappearing. Now, in metaphase, what happens is all the chromosomes that were present within the nuclear membrane, the disappearing nuclear membrane, have now started to line up vertically on the metaphase plate. Now, the metaphase plate is basically a vertical plate or region of the cell. It's usually located in the middle or center of the cell, center line. So that's an important thing to remember here, the metaphase plate. And again, we have centromeres and all that stuff is happening over there. Now, let's move on to anaphase. So in anaphase, what happens is these spindle fibers essentially divide the sister chromatids apart from each other, divide the chromosomes apart from each other. So we have these sister chromatids that these spindle fibers are holding onto on each side of the cell, opposite poles of the cell. And then the last phase, or the second to last phase of mitosis is telophase, where we have almost two separate divided daughter cells, almost. Well, the almost is because we have a cleavage furrow here, or cleavage, we can say, because it's a human body cell. Cleavage, which is basically this area where there's a gap between the two almost divided cells. And so that's what we call cleavage. And that's how we know that this is telophase, is because there is cleavage and there's still a gap between these two almost divided cells. They're not completely divided yet. We have sister chromatids here as well. So next, let's move on to cytokinesis. So in cytokinesis, what happens is the nuclear membrane reforms again, I guess we can say, and then we have ribosomes, other organelles that are formed as a result of these separated daughter cells. So in mitosis, we have a total of total of 2 daughter cells produced. And this is really important differing factor that differentiates mitosis from meiosis, because mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells as results. So now that we've done an explanation of mitosis, let's explore meiosis. Alright, so within meiosis, there's actually 2 big stages. There's meiosis 1 and meiosis 2. But in meiosis 1, we still have mostly the same stages as mitosis, except they're a bit different in terms of the processes that happen. So within prophase 1 of meiosis 1, we have chromosomes, homologous chromosomes, and we have centrosomes, we have spindle fibers still. So those things are similar. And we have here crossing over that already occurred as a result, and thus there are homologous chromosomes here. And that's why you can see the different colors, right? Red and blue here, because the process of crossing over happened, and synapsis also occurs where homologous chromosomes combine. So that's what happens within prophase 1. Also, chromosomes condense, so that's an important part. So in metaphase 1, what happens is we still have the centrosomes. Now the spindle fibers have developed to a point where they are kind of moved to the not the metaphase plate, in that they're moved horizontally, not vertically. So we can't really say the metaphase plate here, but they're moved horizontally along the center line, you can say, of the cell. So we can see pairs of homologous chromosomes are moved to the center line of the cell at a horizontal position due to the spindle fibers pulling them that way. In anaphase 1, we have homologous chromosomes moving to the opposite poles of the cell. So opposite poles, I mean this location and that location, they're both opposite positions or opposite locations within the cell. Thus, we can call them opposite poles. And so you can see homologous chromosomes are kind of separated right there. Now, what happens in telophase 1 is there is a division of cytoplasm, and chromosomes are at the poles of the cells. So what happens is the cytoplasm is dividing, and there's cleavage that forms over there as a result, right? There's this space. They haven't fully divided yet. And so this step is kind of similar to telophase of mitosis, but the process is different. Now in prophase 2, so we're moving on from meiosis 1 here. So meiosis 1 ends there to meiosis 2. So with this, it produces 2 daughter cells. Now, with these 2 daughter cells starts the process of meiosis 2. So meiosis 2 starts with these 2 daughter cells instead of 1 cell that mitosis starts with. And so cell division occurs twice in meiosis, while cell division only occurs once in meiosis. So this is meiosis 2. So in prophase 2, we have new spindle fibers that form. So this is cell 1, cell 2, again, 2 daughter cells. So new spindle fibers start to form, not completely form yet. In metaphase 2, we have chromosomes moving to the metaphase plate, we can say, I guess, because they're vertically aligned in the center of the cell. Right, vertical alignment. And they are held together by spindle fibers there as well. So in anaphase 2, what happens is centromeres completely divide. Sister chromatids move to opposite poles of the cells, as you can see within this diagram of cell 1 and cell 2, what's happening. Then in telophase 2 and cytokinesis 2, we have a reformed nuclear envelope. Basically, the nuclear envelope forms again, and the cytoplasm completely divides. It results in not 1, not 2, not 3, but 4 daughter cells. So meiosis 2 or 2 results in 4 daughter cells instead of 2. So meiosis 1 produces 2 daughter cells. When cell division occurs again, each of those cells gets divided within each step of the process, thus resulting in 4 daughter cells by the end of meiosis 2. So that's why in total meiosis produces as a result 4 daughter cells. Now, let's talk about what are the consequences of cells not passing this stage or having problems with proliferable control or uncontrollable factors that occur through the passing of these stages within the cell cycle. So, several things can result. Firstly, cancer. Cancer can result due to an uncontrollable sense of growth, with cells not listening to growth factor proteins. Now, growth factor proteins are proteins that help contribute to stopping cells from further accomplishing several stages of the cell cycle, either because they haven't done enough work to do so, or because they have some sort of abnormality that is detected within them. To growth factor proteins, cell cycle checkpoints. So cell cycle checkpoints are really important, as these also act as critical kind of checkpoints, you can say, within different stages of the cell cycle. So before a cell passes one stage of the cell cycle, very much checking is done to make sure that, okay, the cell has passed this much of genetic information, or maybe any kind of criteria. Those are what the cell cycle checkpoints check in a cell before it passes on to the next stage of the cell cycle. And obviously proliferably grow. So proliferably grow means uncontrollable growth. So that causes cancer. Now, this is caused mostly by mutation. So a mutation can cause cancer. Then there is apoptosis, which is programmed cell death. This can result if a cell does not pass a checkpoint in the cell cycle. So it automatically kills itself, you can say, because it releases enzymes that, again, produce proteins that eat the cell and the genetic information of the cell. So another important thing here that you can see within AP Bio FRQs, past and maybe future, chromosomal disorders, right? Within this unit, we've learned about meiosis, mitosis, cell division, cell cycle process. So if, things like Down syndrome or trisomy 21, these things are a result of a problem with the processes of independent assortment and crossing over, again, which happen in meiosis as well. So this is really, really important. This may appear on an AP Bio FRQ. It has occurred with at least minimum of one past AP Bio FRQ. So that's a really good thing to make sure that you are confident with. Again, thank you guys for watching. Like, share, and subscribe to not miss any other updates on my channel that I post about AP Biology or other AP course videos. Thank you guys.